Hey everyone, and welcome to This Explainer. I am absolutely thrilled you're tuning in today because we're about to embark on a deep dive into what is literally a biological battlefield. If you're starting entirely from scratch and need to write a really comprehensive, long-form academic answer on antibiotics and bacterial resistance, well, you're in the exact right place. So grab your notebooks, I'm going to be your master tutor today, and we're going to break down exactly how antibiotics work, how bacteria fight back, and the absolutely critical measures we have to take to overcome this escalating crisis. Let's get into it. Okay, so here's our game plan. We've got 5 clear acts. First, our weapons and their mechanisms of action. Second, origins of resistance. Third, the enemy's defenses. Fourth, drivers of resistance. And finally, our new strategy for prevention. All right, let's jump right into section 1, our weapons and their mechanisms of action. Now, before we look at specific drugs, we really need to understand the golden rule of treating infections. It's a concept called selective toxicity. You see, these drugs have to be highly toxic to the invading bacteria, but completely harmless to you, the human host. To pull that off, we have to exploit the biochemical differences between the pathogen and the host. We're targeting parts of the bacterial cell that humans just plain don't have. When we deploy these selectively toxic weapons, they generally fall into one of two camps. First, we have bactericidal drugs. These are the assassins, right? They kill bacteria directly. Penicillins are your classic example here. On the flip side, we have bacteriostatic drugs. Now, these don't kill the bacteria outright. Instead, they prevent them from growing and replicating. Tetracyclines do this. This distinction is foundational, so definitely note it down. But remember, the ultimate clinical success usually depends on the patient's own immune system swooping in to mop up those weakened bacteria. What's super fascinating is actually mapping out these specific biochemical targets. Take a look at this. Sulfonamides, like sulfadiazine, they disrupt bacterial folate synthesis. B-lactomes and glycopeptides, like penicillins and vancomycin, they go right after the bacterial cell wall, which is brilliant because humans don't even have cell walls. Polymyxins disrupt cell membranes, and chloramphenicol binds to the 50S ribosomal subunit to just entirely halt protein synthesis. You'll definitely want to memorize a couple of these for your exam. It's a perfect illustration of our varied attack strategies. Basically, if we can't break down their walls, we starve them of proteins or totally wreck their metabolism. Moving right along to section 2, the origins of bacterial resistance. Before we start pointing fingers at modern medicine, we actually need to look way, way back into deep time. Get this, researchers sequenced DNA from a deep cave system in New Mexico that had been completely isolated from humans for over 4 million years. And inside, they found a species of bacteria called Panibacillus that was already resistant to most of our modern clinically used antibiotics. Crazy, right? This points to something called the resistome, this massive pool of ancient resistance genes. Remember, a lot of our antibiotics originally come from soil-dwelling fungi and bacteria. For literally millions of years, these microorganisms have been fighting a microscopic turf war in the dirt, evolving natural, innate defenses against the exact same chemicals we use today as drugs. So, if these defenses are millions of years old, well, that begs a vital question for your essay. How do bacteria actually share these defenses? How on earth does a resistance gene get from a harmless little bug in the soil into a dangerous superpathogen in a modern hospital? They do it through some seriously incredible genetic shuffling. Make sure you write these 3 mechanisms down. First up, plasmids. Think of these as tiny circular pieces of extra DNA that bacteria can easily pass around to one another, almost like trading cards. Second, transposons. We call these jumping genes. They're mobile elements that can literally cut and paste resistance traits right into the bacterial DNA. And third, gene amplification. When bacteria are put under stress, they can just duplicate their pre-existing resistance genes, pumping out more and more defensive enzymes. This is the exact genetic blueprint for how resistance spreads like wildfire. Okay, so we've got the genetics down. Let's hit section 3, the enemy's defenses. When those resistance genes are activated, what's actually happening down at the cellular level? How are they physically stopping our drugs? Well, it boils down to 3 main biochemical workarounds. The crucial point here is that bacteria physically adapt their structure. Number 1, they alter the drug binding sites. Simply put, if the drug can't dock, it can't kill. We see this a lot with penicillins. Number 2, they reduce drug uptake. They might literally close off their pores or act like a bouncer and actively pump the drug right back out, which happens all the time with tetracyclines. And number 3, they alter enzyme sensitivity. They tweak their internal machinery so the antibiotic just doesn't affect them anymore. It is cellular warfare at its absolute finest.
Which brings us to Section 4, drivers of resistance.
We know the ancient genetics and we know the biochemistry, but why is this suddenly a massive global public health crisis right now? Well, according to policy documents from the Indian Council of Medical Research, it's us. Severe anthropogenic, meaning human-driven, factors are creating immense selection pressure. When we use antibiotics improperly, we're just killing off the weak bacteria and leaving only the strongest, most resistant ones behind to multiply. This happens through the irrational prescription of broad-spectrum antibiotics when they really aren't needed. It happens through poor regulation, like over-the-counter sales that let people self-medicate for a simple viral cold, which antibiotics don't even treat. And massively, it happens in agriculture, where antibiotics are pumped into livestock not to cure sick animals, but just to make them grow faster. And all of this misuse has led us to a frankly terrifying statistic. Just 2 years. On average, resistance normally appears within just 2 years of the introduction of a brand new antimicrobial agent. We are burning through our pharmacological arsenal at a totally unsustainable rate. That sobering reality leads us right into our final act, Section 5, our new strategy, prevention. We simply cannot invent our way out of this anymore. New drug development has really stalled out, so we need systemic public health solutions. Recognizing this, major health bodies like the Indian Council of Medical Research have pivoted toward a critical concept that you absolutely must include in your answer, the Antimicrobial Stewardship Program, or AMSP. It sounds fancy, but the core goal is just to ensure the rational, evidence-based use of the drugs we currently have so we can ensure their longevity. This philosophy builds into a super clear, actionable, 3-step implementation strategy for hospitals. This is very testable, guys. Step 1 is implementing that effective clinical stewardship we just mentioned. Step 2 is remarkably simple but absolutely crucial. Meticulous hand hygiene in healthcare settings to physically stop the bugs from spreading. And step 3 is the continuous surveillance and monitoring of multi-drug resistant organisms so we know exactly what the enemy is doing in real time. To actually enforce this stewardship, hospitals have to focus heavily on education and regulation. Doctors need solid, evidence-based training on prescribing the exact right drug, dose, and duration. We need optimization campaigns to make sure everyone understands the dangers of misuse. And crucially, there has to be strict pharmacovigilance and prescription audits. Basically, doctors must be held accountable for what they're prescribing to help restrain the overuse of these incredibly precious resources. As the Indian Council of Medical Research so perfectly summarized it in their policy document, the solution to the current approaches to antimicrobial resistance is to preserve the effectiveness of the drugs which are presently available. Right now, conservation is our single most powerful medical tool. We've got to protect the arsenal we already possess. So, as you wrap up your essay notes and your studying today, I want to leave you with a final thought to really ponder, knowing that these resistance genes are millions of years old, that they are constantly adapting, and frankly, just waiting for us to make a mistake. What role will you play in antimicrobial stewardship to protect the few weapons we have left? Thank you so much for joining me in this explainer. Keep this 5-part framework in mind, study hard, and you are going to absolutely crush this topic. See you next time.